for this class of diseases. Then I'll move into the most commonly used MR biomarkers for SCAs. And there are three modalities that I will focus on, starting out with volumetric markers. This is structural MRI. Then moving on to microstructural markers obtained with diffusion MRI, and finishing up with biochemical markers using MR spectroscopy. And I'm going to be doing more of a deep dive into MR spectroscopy because that is the modality that I more or less dedicated my career on, and it's less frequently used than the other modalities that I will be focusing on. And finally, I'm going to finish up with an update on the status of where we are with clinical trial readiness with respect to MR biomarkers in SCAs. So before I get going, I'd like to find out the professional background of our audience. So please choose the option that best represents you in the first question.
Okay, already 70% have answered. We will wait a couple of seconds more and then I will show the results. Okay, so here are the results.
Okay, so we have a third of our audience made up from neurologists, and then good representation of neurology residents and neuropediatricians, psychiatry, geneticists, psychologists, and we also have some patient representatives. That's great. Thank you. Thank you, Sonia.
Mm-hmm.
Okay. So let's start out with a very short background on SCAs. I'm assuming this audience does not need this background, but nonetheless, here it goes. So these are dominantly inherited movement disorders, and there are more than 40 genetic forms. They cause neurodegeneration in the cerebellum primarily, and many of them also cause neurodegeneration in the brainstem, as shown here with a flattening of the pons in SCA1, as well as the cerebellar atrophy that's quite visible in this T2 weighted MR image. This neurodegeneration results in progressive loss of motor coordination, affecting gaze, speech, gait, and balance. The most common forms are SCA1, 2, 3, and 6, and they all happen to be polyglutamine diseases. And there are currently no disease-modifying therapies for any of the SCAs. So I'd like to start out with going over what is MRI typically used for in the clinic. And conventional MRI, structural MRI, is the modality of choice, and it is very frequently used in diagnosis to confirm cerebellar degeneration and to also rule out other potential causes for ataxia. So this figure here, this image here, is from a recent review that I collaborated on with Drs. Ian Harding and Catherine Reitz. And it shows you, first of all, the cerebellar degeneration, and in many of the forms, the brainstem, in particular, the pons degeneration. But it also shows you that there is some genotype specificity. The atrophy pattern is not the same between different ataxias. So on the top row, I have the most common SCAs, and the bottom row, other hereditary and sporadic causes of ataxias. And if you look at the SCAs, for example, you can see in SCA6 there's quite a lot of degeneration, atrophy of the cerebellum, but the brainstem, the pons, is not nearly as much affected as, for example, in SCA2, where you do see this flattening of the pons. And then why do we need new outcome measures in SCAs? And that's primarily to be ready for upcoming clinical trials. There's a lot of work. I'm going to go through a little bit of that, a lot of work gearing up towards large clinical trials in ataxias, but we need to do some more work in terms of outcome measures. The primary outcome measure is a clinical one. It's called the Scale for the Assessment and Rating of Ataxia, the SARA scale. It consists of 8 items and evaluates gait, stance, sitting, speech, and limb kinetic functions. And it's by far the most widely tested, used, and validated clinical outcome measure for SCAs, especially the EuroSCA group has done a lot of studies to validate it in the multi-site setting. And these results here are from one of their papers by Dr. Heike Jacobi under the leadership of Dr. Clockgetter. And what they did here was calculate the sample size needed per group in a 2-group interventional trial of 1 year duration. So this is not a short trial, for various effect sizes in the most common SCAs. And if you look at, let's look at the SCA1 panel, for example, for an effect size of 30%, which is pretty good effect size, you need like 200 or more participants per arm. In a rare disease, this becomes very problematic. In a different paper, these data were combined with predictions based on a United States study. That's the CRC-SCA. This is the Clinical Research Consortium for the study of SCAs. And in this plot, you see that based on the US data, the numbers are even higher. So that means we need additional outcome measures to supplement the clinical measures and reduce these sample sizes to make these upcoming trials viable and feasible. So now I'm going to start out with neuroimaging, and I'd like to know if you use quantitative neuroimaging in your research or practice. And if you do, please select one of these choices. Which modality you use.
Okay, 15 seconds left and I will show the results. Okay, so right, here are they.
Okay, most people use volumetric MRI, then there's a good number who use diffusion MRI. Okay, even MR spectroscopy, glad to see that. Some perfusion, functional, okay, this is kind of my expectation, but it looks like only about 20% of the audience does not use neuroimaging. The majority does. Okay, thank you, Sanya.
You're welcome.
Okay, let's move on. So, okay, after the quiz.
Gülin, maybe just click again on your slide and then with the arrow to move forward. Yeah.
Okay, this one worked after multiple trials. Okay, so this is again a figure from the review that I referenced with Ian Harding and Catherine Reeds. And here we identified the most commonly used quantitative imaging, quantitative MR methods in ataxia. Actually, I can't say quantitative imaging because PET spec type nuclear imaging modalities are used, but not nearly as commonly. And here we are focusing on the most common form of recessive ataxias, Friedreich's, most common form of autosomal dominant ataxias, this is SCA3, and most common sporadic as MSAC. And for all those cases, structural MRI is used more, also confirmed by our audience, followed by diffusion MRI and followed by spectroscopy. So that is what we have focused our review on, and that's going to be the focus of my talk today. So starting out with volumetric MRI, there's substantial amount of work out there. I'm only going to highlight a few studies to make different quantitative points. So the first one is about the genotype specificity of the atrophy patterns in SCAs. And this is a very classic paper by now by Dr. Reeds, came out in 2013, where they studied patients with SCA1, 3, and 6 longitudinally with a follow-up, and they had well-matched control groups for each of these 3 SCA groups. And if you look at these panels, these are regional volumetric assessments, and the volumes are normalized by the total intracranial volume. You'll see, for example, if you look at the brainstem, SCA1 and SCA3 show severe atrophy and not so much SCA6, and here are the control groups. Or when you alternatively look at the cerebellum, all 3 groups are distinguished pretty well in terms of the extent of atrophy in the cerebellum. So the atrophy patterns differ from genotype to genotype. And this was also one of the first, well, actually, I think in this case, the first study that showed that MRI measures, these volumetric measures, were more sensitive to change than clinical decline as measured by the SARA score. So here we are looking at effect sizes, which are mean annual percent changes divided by the standard deviation of annual percent change. First, comparing the clinical scores that they utilized in the study, and essentially showing SARA is, in general, the most sensitive for these genotypes to change. And then looking at the same effect sizes, the standardized response means of volume loss rates, they observed that the effect sizes obtained by these volumetric measures were in all cases higher than the clinical scales. So here, brainstem, for example, minus 1.5, minus 1.6 versus Pons minus 1.5. So that's 1.5 times standard deviation lower for the change over time. So, and then same with SCA3, SCA6. So effect sizes are always higher for these volumetric measures, showing higher sensitivity to change. So then the question is, how fast are these volumetric reductions detectable? How soon does the rate of change become different than 0? And how soon can this be reliably measured? This is from a recent study that just came out last year. It's a collaboration with Dr. Pagnopoulos and Tim Kostrick at the University of Iowa. And they analyzed with sophisticated volumetric methodology, mostly in-house developed, longitudinal data obtained from patients with SCA1. These are T1 weighted images, and patients were followed for up to 3 years. And they identified the volumes that were most sensitive to change. And then they modeled those data to determine which volumes were most sensitive to change, like the last study, and then how fast these changes were reliably detected. The Pons volume was the most sensitive to change, and the accelerating putamen volume as shown here. And in this plot, we are looking at detectable delta volume, detectable change in volume. And these are the estimated times for this change to be reliably detected. So you see pons in about half year, in 6 months, it should be reliably detected. Then cerebellar lobule 6, cerebellar white matter, deep nuclei, and putamen, going up to 7, 8 months. So this is important because this would enable shorter trials. Then moving on to an example, this time from Brazil, and in SCA3, this came out a few years ago. And here, one of the focuses of the study was the detectability of these changes at the premanifest stage. So this was a pretty large study in terms of sample size, and they grouped their subjects into non-ataxic mutation carriers, the premanifest group, and then they divided them up according to time from ataxia onset. This is late stage ataxia, this is early stage ataxia. And as you can see, the volumetric changes increase with time from onset, but most importantly, even the non-ataxic mutation carriers show some atrophy, primarily in brainstem, some atrophy in substantia nigra, and it's not shown here, but they also saw spinal cord atrophy at this stage, which is quite important. And from this, in the same study, we move on to microstructural damage in SCA3. And this is their entire group, and showing the classic DTI parameters that they extracted from their cohort with fractional anisotropy here and all the 3 diffusivities, axial, radial, and mean diffusivity. And you can see there's quite widespread involvement of white matter. And there's actually quite a lot of work of diffusion MRI showing widespread white matter involvement in various SCAs. This is one of the recent examples. One missing piece in literature in diffusion MRI has been that there have not been too many longitudinal studies. Most studies published were cross-sectional. So this is a recent example from a diffusion study. This is from our group, where there was a small longitudinal component. This was led by Drs. Park and my dear colleague, Dr. Christoph Langlet. So here we are again back to the theme of genotype-specific microstructural damage, and we are looking at the most common SCAs 1, 2, 3, 6, and you can see with the FA here, higher diffusivity, lower FA, you see it in all genotypes, but by far the most involvement is seen in SCA2 and the least in SCA6. This is similar to volumetric results. And then moving on to the longitudinal component in the study. So these are plots showing data from individual study participants. In red are all the individuals that had SARA scores higher than 2.5, which is considered manifest ataxia. SARA score lower than 3 is considered pre-manifest stage of disease. So in blue, we have the pre-manifest individuals. So first of all, you see the expected increase in the SARA score in everyone, except for maybe this participant who's stayed at the pre-manifest stage throughout the study. And then there are these 3 individuals who started out the study at the pre-manifest stage, and 2 of them started showing ataxia symptoms during the study. Now, if we look at one of the measures, the axial diffusivity at the middle cerebellar peduncle, First of all, interestingly, you see that those two premanifest participants already at baseline, when they were at premanifest stage, they were already quite well separated from controls in black here. And then you can see the trend for increase, the progression of axial diffusivity. And this individual never reached the manifest patient range, but you still see the increase. So this shows sensitivity to alterations even before ataxia onset. And then I'm going to switch on to spectroscopy, and I have another questionnaire here. Do you use MRS in your research or practice? If not, why not?
Okay, another 15 seconds and I will show the results. Okay, so here are the results.
Okay, let's see. Well, over 20% of the people are using it all the time. So among those who do not use it, it is because it does not easily fit into the clinical workflow. Okay, that makes a lot of sense. And then it is too difficult to acquire good data. Yep. And MRS acquisition methods are not standardized. So those are the following two top reasons. And there are other reasons why people don't use them, the quarter of the audience. Okay, thank you. This is very helpful. All right, let's see now. Will we be able to, okay, next. Oh, all right. Gets hung up somehow after the questionnaires. So first, a little bit of background, very broad background on high field MRS. So this is how the data looks like, the MR spectroscopy data. Everything I will talk about today is on single voxel spectroscopy, where you select a volume of interest like this and collect the data. You suppress the water signal so you can access these millimolar concentration metabolites. And each of these peaks comes from a different neurochemical. So different neurochemicals based on their structure have signatures in the spectra. Let's talk a little bit about the largest peaks. For example, this one here, total N-acetyl aspartate, NAA, is by far the most widely validated MRS marker for the brain. And it is considered an indicator of neuronal health. If you have neuronal dysfunction or loss, this peak goes down. Then we have total creatine here at 3 ppm. And that one is in general considered an energy metabolite, but also can represent gliosis. Increases in total creatine can represent gliosis. Then we have total choline, which increases in demyelination. And we have myonosto over here, and that is considered a putative gliosis marker. So we collect the data, we do a mathematical fit, and get concentration information on a series of metabolites that include not only the major ones that I talked about, but also neurotransmitters like glutamate, GABA, and antioxidants like vitamin C and glutathione. To summarize the cellular and biochemical processes measurable by MRS, first you have indicators of changes in cell number, viability, neuronal markers are NAA and glutamate. glial markers, mannostol and glutamine, which is primarily localized in glial cells. Then there are demyelination and membrane turnover markers, choline, and mobile lipids that become visible in some conditions. Defects in energy metabolism, creatine, phosphocreatine, lactate, glucose, and oxidative stress with glutathione and vitamin C. One thing to remember when you think about spectroscopy is that neurochemical profiles and these spectral patterns are region dependent. If you look at the ratios of the major peaks here, they are different from region to region, depending on the cellular composition of that volume of interest. So going back to how spectroscopy has been used to study SCAs, this is one of our earlier studies showing the sensitivity to neurochemical alterations in individual subjects. I'm showing data here from a control individual and from a patient with SCA1, and these spectra are acquired from the top ones from cerebellum and the bottom from the pons. If you compare, just visually compare the spectra from the control versus the individual with SCA1, you can see the NAA is lower, creatine is higher, and mannostol is higher. This is even without doing any fitting and quantification. By visual assessment, you can do this. And same story for the pons. These markers have been shown to correlate with the SARA score. So here, if we look at the NAA panel, this is the neuronal marker. The higher, the lower the SARA score, the higher the NAA. So this is non-ataxic individuals, and with the increased ataxia, the SARA score increases, goes up to 40. So these are relatively early stage individuals. Same story with glutamate, and mannostol is the opposite because that is the gliosis marker. So the higher the SARA score, the higher the mannostol. I'm going to digress a little bit and show you some validation data that we have done in mouse models of ataxias to understand what these markers represent in terms of pathology. So this is from one of our earlier studies with Drs. Orr and Clark, where we studied mice longitudinally and at different time points sacrificed subgroups of mice and compared the MRS findings to the histology findings. One thing to show here, again, by visual assessment, on the top, I have wild type mouse. These are data from one mouse followed up to 1 year. Pretty stable spectral pattern. When you look at the SCA1 mouse, you can, again, visually see the drop of NAA over time, clearly, an increase of mannostol over time. When we looked at the correlations of the MR markers with molecular layer thickness, this is one of the reliable pathologic quantitative measures in this model, we saw good correlations like we had seen with the SARA score. So the higher the molecular layer thickness, the higher the NAA and glutamate, and the lower the mannostol. In a different mouse model line, we have seen that these neurochemical changes appear even before gross pathology. So this different model developed by Huda Zogby's lab does not show gross pathology up to here 39 weeks of age. So these are the pathological measures. But throughout that time frame, there are a lot of biochemical changes. Let's look at the bottom panels here. For example, glutamine and creatine are higher in the SCA1 mice than the wild type litter mates. So, and note that these are different markers. These are other biochemical markers that designate the earliest pathological, or reflect the earliest pathological changes, and not the degenerative markers that were primarily NAA, glutamate, and myonostal. Then the big question, of course, for clinical trial use is MRS sensitive to disease reversal? We addressed that using some conditional SCA1 mice where the expression of the mutant ataxin-1 gene is under doxycycline control. And in pathology, you see if you treat these mice with doxycycline, the molecular layer thickness is preserved, whereas in the untreated mice, it is thinner. And my collaborators had shown that the neurological phenotype and pathology are reversible at early to mid stages in this model. So we did a study where we compared spectroscopic markers with invasive measures, starting out with quantitative PCR that shows you how well the doxycycline suppressed the transgene expression. So here I'm showing receiver operator characteristic curve, a ROC curve, and an area under the curve of 1 in this plot indicates there is no overlap between the two groups. Treated versus untreated groups are separated perfectly. So this is almost perfect separation with area under the curve of 0.97. Same with the molecular layer thickness, and same with a combined measure, MRS measure made up of NAA, myonostal, and taurine. So MRS in this case was as sensitive as the invasive outcome measures to distinguish treated from untreated animals. We also looked at motor testing. Rotarod is the classic method to evaluate balance in these animals, and it did separate the treated from untreated groups, but not with as high sensitivity. Going back to patient work, of course, the question is, are these markers sensitive to change, sensitive to disease progression? And this is a study that came out a couple of years ago where we compared the sensitivity of volumetric MR measures and spectroscopic measures, as well as SARA, the clinical evaluation. And these are, again, spectra from individuals from 3 different regions: vermis, cerebellar white matter, and pons. And you can, again, see the reduction in NAA, elevation in myonostal, and in this case, the elevation of creatine relative to controls by visual inspection of the data. And these are, this panel shows the volumes that were evaluated in this study from structural MRI. We saw that both MRI and MRS were sensitive to change over time to disease progression, and these are the MRS metrics and volumetric MRI metrics that were most sensitive to change, with % change per year values. And in the case of MRS, NA over myonostal ratio, a neuroglial ratio in the pons was found to be most sensitive. And for volumetric MRI, consistent with prior reports, pons volume was the most sensitive to change. And again, if you look at the effect sizes for controls and for SCA1 group, pons volume, the volumetric measure, was most sensitive to change, followed by NA over myonostal in the pons, followed by the SARA score. So let's do a quick test of what you have learned so far in spectroscopy. So I have three spectra here acquired from a volume of interest like this from the cerebellar white matter. And 2 of these are from SCA-mutation carriers and 1 from a healthy control individual. So which are from mutation carriers, 1 and 2, or 2 and 3, or 1 and 3? Sanya, if you can bring up the question.
Yes, I have just right now launched the question. Okay, still 10 seconds to respond to this question, and then I will show the results. Okay.
All right, congratulations. 80% of you were able to get the correct response. And again, I'm having trouble with this. Okay. And you, in fact, identified spectra obtained from 2 pre-manifest individuals. On the left, we have a pre-manifest stage individual with SCA1, and on the right, we have an individual with SCA3. So very clearly, NA is down, creatine is up here, and minostol in the case of SCA2 is up relative to the control individual. So it shows you even by just looking at spectra, you can identify a pre-manifest individual, distinguish a pre-manifest individual from control, just on the basis of one spectrum. And those data came from a study from a couple of years ago, where we looked at the classification accuracy of MR spectroscopy markers, and the control versus manifest individual separation was pretty clear. This is an analysis, statistical machine learning algorithm called distance-weighted discrimination. And we saw that the groups were very clearly distinguished, which is what we had expected, but we also had 9 pre-manifest mutation carriers in this study. And we noticed that most of them were classified with the SCA group based on their spectroscopy scores that this analysis gave us. But few of them, 3 of them, were not classified with SCA. So we asked the question, for example, this individual here, individuals at pre-manifest stage have the circle around the data point. We asked what was different about them, and we calculated their estimated years to onset. And we saw that all those 3 individuals had estimated years to onset that are longer than 10 years. So the closer you are to disease onset, the higher the chances of detecting these neurochemical changes, which makes a lot of sense. But this also tells you that these changes are there up to 10 years before ataxia onset. And here I'd like to go a little bit towards the technical side and emphasize the importance of data quality when you do spectroscopy. Things like signal-to-noise ratio, a flat baseline, good water suppression, lack of unwanted coherences, contaminations, and good spectral resolution are very, very critical to get reliable quantitative information from spectra. And we systematically evaluated this. Some of you responded that there are no, acquiring good quality data is difficult for spectroscopy in the last questionnaire. And that is because the methods that are provided on the scanners, the conventional MRS protocols, are not very highly optimized. And in this study, we systematically evaluated that in the same subjects by acquiring data with the conventional protocol and the advanced protocol in a randomized fashion by MR technologists, and showed that the reproducibility with the advanced protocol was much higher. If these are data overlaid from 30 individuals, and you can see there's a lot more variability with the conventional protocol. And when you use this kind of optimized methodology, the reproducibility can be quite high. Here I'm showing some spectra that are acquired from one subject 4 times with weekly intervals, and all the cerebellum spectra here acquired at 4 Tesla are overlaid. So you can see how well they overlay, how reproducible the spectral pattern can be. And you can collect this kind of high quality data as long as such advanced methods are used at all different sites. So these are data showing the reproducibility between sites across 3 institutions, ours, Paris, the ICM group, and at MGH, Harvard. So these are cerebellum spectra collected at the clinical Siemens 3T scanner. And we did a quantitative analysis with our colleagues at ICM, Dr. Fanny Moschel, Alexandra Dur, and showed that the neurochemical profiles that you can obtain from such spectra are identical. These are different healthy volunteers, different operators. The mean concentrations that you obtain from the full neurochemical profile are highly consistent. So to summarize just the spectroscopy part before I move on to the final chapter of the talk, MRS is sensitive to premanifest alterations, disease progression, and in animal models, we have shown that abnormalities are detected prior to gross pathology. The reversal of pathology was detected again in animal models, but spectral quality is absolutely critical for good reproducibility. The good news is the high spectral quality can be obtained on standard 3 Tesla hardware using advanced acquisition methods. So finally, the clinical trial readiness aspect. There are a lot of potential treatments for SCAs in the pipeline, and they go from the DNA level to RNA and protein level, and even targeting some downstream pathways. So there is a real need, an urgent need for additional outcome measures for these upcoming trials. As far as MR biomarkers go, this is where we are. This is a summary plot, again, from our recent review, showing approximately where we are with different MR modalities, MR biomarkers, starting with morphometry, structural connectivity, diffusion, functional connectivity, and neurochemistry. So the first phase is biomarker discovery, followed by analytical validation and clinical validation. Morphometric measures, as I said, they have been validated in multi-site prospective studies with large cohorts, and a lot has been done. So we know they will be useful in upcoming clinical trials. Neurochemistry, there has been a lot of validation work, and more now we are getting into multi-site validation studies. And structural connectivity, more longitudinal studies are needed at this point, but there are some multi-site efforts from the European SCA group, as well as US-Europe collaborations. And just to summarize the utility of these MR biomarkers, the current idea is that they will be useful at the enrollment stage to stratify enrollment cohorts, for example, by looking at early changes in the brain, in microstructure, in chemistry, as shown here, as well as in the follow-up stage to monitor treatment responses. Is the atrophy slowed down with the treatment? are the neurochemical abnormalities reversed? Are the microstructural abnormalities reversed? And then one current ongoing big effort towards that is this SCA study that focuses on SCA1 and SCA3. It's a multi-site study funded by NIH in the US, and these are all the different sites involved. And we have two sites in Europe too, with Dr. Clark-Getter in Bonn and Dr. Alexandre Dur in Paris. And we have 6 imaging sites, 4 of them in the US and 2 in Europe. And we are collecting natural history data up to 3 years, starting out from baseline 6 months, 1 year, 2 year, 3 years. And those data are currently being collected, and the first results will be coming out pretty soon. And it uses 3 Tesla platform, morphometry, diffusion MRI, MRS, and we also have resting state as a more exploratory modality in this study. So the key points are morphometric MR, diffusion MRI, and MRS are expected to be powerful outcome measures in upcoming SCA trials, both for participant stratification and treatment monitoring. And morphometric measures have been most widely validated in multi-site trial settings, and they are highly sensitive to progression over time. And the diffusion MRI and MRS promise high sensitivity, especially at the premanifest stage. And ongoing longitudinal multi-site studies with premanifest and early stage participants will show us the predictive value of MRI, MRS markers. So far, most of our data are still, for that question, are still at preliminary data stage. I'd like to acknowledge our ataxia imaging team at our center. Doctors Christophe Langlet and Pierre-Gilles Henry have been leading Friedreich's ataxia, large Friedreich's ataxia multi-site efforts, in addition to all the SCA work we've been doing with our group. And we've been working with a lot of partners over the years on both the clinical application of our methods and starting out from development, validation, the technical improvements, and a lot of automation we have done over the years. Our clinical collaborators, more spectroscopy collaborators are here, our local collaborators, past members of the lab, and the funding sources. And thank you for your attention, and I'm looking forward to your questions.
Yeah, thank you, Gülin. Thank you for this very clear and insightful presentation and summarizing all of your findings and results. I see there are already 2 questions or 1 comment. The first is just a comment. Getting insurance to pay for MRS is difficult. Mm-hmm. And the next one is, were there any pre-manifest changes in glutathione in either the mouse models or patients with SCA?
The second one is easier. We haven't seen any pre-manifest changes in glutathione so far. And the first question about insurance, that is true. Although my understanding is that is much more of a problem in the US. We wrote a consensus paper on the value of MR spectroscopy for CNS disorders. It was a global effort. And through those discussions, it became clear that a lot of institutions in Europe actually have a lot easier time getting insurance reimbursements to add spectroscopy to their clinical protocols. But I think that primarily involves brain cancers. So tumors, that's an indication where insurance seems to be, insurance reimbursement seems to be a lot more straightforward. Other indications are more difficult, and that is accurate.
Mm-hmm. So I don't see any other questions. There was somebody who raised his hand. I think you can just write your question if it was about something you wanted to ask. Otherwise, we can wait just some minute. Maybe other questions will arrive, and otherwise we can conclude here. For the CTs, is there a consensus protocol for the use of MRS?
So there are a couple of consensus papers in literature for general use of MRS. There's a 2014 paper that came out in Radiology that was in one of my slides. So, and then a technical consensus paper came out in MRM by Martin Wilson et al. These had both 50 experts from around the world came together and put down their guidelines for data acquisition, analysis, interpretation. And this year, well, I think I should say 2020, not this year, last year, NMR Biomedicine had a special issue on advanced spectroscopy in each chapter addressing different aspects of MRS data acquisition and analysis. There is one paper there that I led with recommendations for advanced MRS. Now that's for general MRS use. For ataxias, we are currently working in a under Ataxia Global Initiative. We have working groups. One of them focuses on MR biomarkers, and a consensus paper will be coming out of that to providing guidelines for data acquisition for all these different MR modalities by experts in the ataxia field about how the data should be acquired and with example acquisition parameters. So that's coming geared towards ataxias.
Mm-hmm. Okay, thank you. Yeah, and greetings from Bart van der Warenborg. Wonderful talk, and thank you.
Thank you.
Then another comment from Alan from Ataxia and me. Thank you for this interesting presentation. As a patient and a patient advocate, it's sometimes hard to involve patients in such high-level research information. Just a comment, maybe?
That's true. I would recommend some webinars that patient advocacy organizations have been organizing, especially during the COVID time. The number of those webinars that make this information more accessible increased. So check out National Ataxia Foundation and the UK Ataxia organization, and webinars like these, although this one still is fairly technical, but there are a lot of other webinars out there by advocacy organizations I would recommend.
Mm-hmm. There is another one. Can one compare volumetric MRI that were acquired with different methods?
Yes, that is more straightforward for volumetric MRI, even if the acquisition parameters were different, the data were acquired on different vendors. But your variability, quantitative variability, will be higher the more variability you have in the acquisition methods. So it's still ideal to try to harmonize the data acquisition, but you can get very useful information even if that is not the case. There's an Enigma Ataxia effort that Dr. Ian Harding is leading from Monash from Australia, where they gathered data, structural imaging data that were collected under all sorts of different circumstances and on different MR scanners, and they are getting very good data. But there you need really large sample sizes, large volumes of data, the higher the variability.
Right. Okay. May I know what is being done regarding structural and functional connectivity?
So I a little bit touched upon the diffusion MR work that's out there, that's structural connectivities, that is now part of several multi-site efforts. There's the ESMI, the European SCA-3 multi-site study that's doing diffusion MRI. Already SCA has a diffusion MRI. And actually functional connectivity, functional MRI is part of those protocols too. We definitely need more longitudinal data that are now being acquired with those modalities.
Mm-hmm. Thank you for your work and very clear presentation about such issue. Do you correlate also your interesting results with any lab biochemical markers?
That's a really good question. That kind of work is also in the works. For example, NFL measured either in CSF or more commonly now in blood as a neuronal marker is being collected more and more routinely. So we have plans to correlate our MR measures with the blood measures. So of course, if you can get a very good validated high sensitivity marker just with a in a blood sample, that makes life a lot easier than laying in the scanner for an hour and motionless. So that kind of work is ongoing, yes.
Mm-hmm. And another question from the same participant. We have retrospective 1.5T MRA patients results. Is it still be possible to gain some data for the future research?
I get if the question is, can those data be utilized for future research? I would say yes. The initiative that I mentioned, the Ataxia Global Initiative, and if you just do a Google search, you will find it, learn more about it. There's a nice website. So in that initiative, under that initiative, we are working towards providing guidelines for data acquisition. And as part of that initiative, the ultimate goal is to establish an ataxia database, imaging database. And contributions to that database, we are hoping will come from global partners. So we are working towards that because that kind of centralized effort is very, very needed and will be valuable. So we are working towards that right now, gathering data through those global initiatives. Okay, so Sanya put the website in the chat.
Yes, exactly. So everybody can visit it and take a look on interesting parts of it. Yeah, so I think we, yeah, thank you by Lucy.
Thank you for your interest and all your questions.
Okay, so I don't see any other questions. So thank you again, Gülin, for your presentation, for being with us here today. And there is another webinar next week. So stay tuned. You can subscribe through our website, ERN-RND. Stay safe. Thank you, everybody, and goodbye.
Take care, everyone.